Hello, welcome to Science Hangout from Science Mission. I'm Sadashiva Pai. We are going to start our second Hangout today. We have today Dr. John Kirby, Professor of Microbiology at the University of Iowa with us. Welcome, Doctor.
Thank you. It's nice to be here. Thanks for the invitation.
So Dr. Kirby is an expert in molecular microbial signaling. We'll hear from him very soon. Before that, let's look at some of Dr. Kirby's achievements. I'm going to highlight only a few of them, as it may take more than an hour to go through all his honors and accomplishments.
Right. The list was quite an exaggeration on my part.
That's fine. Dr. Kirby is Director of Residence Research in Urology. He is the Chair of Graduate Admissions in Microbiology. He's a Program Director, Sloan Center for Exemplary Mentoring. He's a member of interdisciplinary programs in informatics and genetics. Dr. Kirby held Division Chair Advisor position at American Society for Microbiology. He also instructed advanced bacterial genetics course at Cold Spring Harbor Laboratory. Dr. Kirby is the science advisor for Hennepin Life Sciences. He's an academic editor for PLOS ONE. Dr. Kirby is the editorial advisory board of Molecular Microbiology, Advanced Bacterial Genetics, and he is also chair of Gordon Research Conference on Sensory Transduction in Microorganisms. He is a reviewer for more than 40 scientific journals. He has received Outstanding Teaching Award for many years. Dr. Kirby has written several review articles and book chapters, and his research has been highlighted over several years by well-known publications like New York Times, Nature Reviews, Biotechnics, etc. He has also been highlighted in the local TVs too. Now let's hear from Dr. Kirby. Professor, can you tell us about yourself and your background, education, research projects you are working on right now?
Sure. Again, well, thanks again for the invitation. Thanks for listing those things. I realize that I've agreed to do too much. That's for sure. Yeah. So basically, I'm a molecular microbiologist, as you pointed out. Our main focus is thinking about microbial communities, and I will have a lot to say about that. That's how we got into doing the microbiome. But I started out studying soil bacteria, and that's essentially laid the foundation that allowed us to make the transition into studying human microbiome. So my background really started out in chemistry. I was informed basically from growing up in the upper Midwest. Lived in a small town outside of town, surrounded by farms on all sides. We weren't farmers. My dad was a pharmacist though, and so growing up, I was thinking about molecules, and that's really what laid the foundation for me. I didn't realize that I was being indoctrinated as a chemist until much later, until I got to college and I looked back on what had happened. So I went to the University of Illinois. I started out as a chemical engineer, switched into chemistry, eventually switched into biochemistry, and then eventually became a microbiologist. And so really I've had a lot of cross-training, if you will. I think I benefited from that in a great part because by being on the engineering side of things, I had to take a lot of math and a lot of physics and a lot of the coursework that I wouldn't have taken if I'd started out as a microbiology major, for example. And so that has helped me do some of the things that I do now because I can tap into that background and think more broadly about some things.
That is interesting.
So that's really where my background got me pointing into this direction. And then, like I said, since I grew up in a rural community, Princeton, Illinois, I was surrounded by nature, and I thought about what was going on in the environment when I was a kid, and I asked crazy questions that kids ask, and I got good feedback from my parents and teachers all the way along, and so I was not discouraged from doing that kind of inquiry, and so that's really why I'm doing what I'm doing today.
That's interesting. So what made you like? I mean, like you mentioned about the soil and your connection with the earth. So, so what made you really think eventually I need to go to microbiology than the biochemistry or other aspects of life sciences?
Well, that's a good question, and that's luck. That's serendipity. I went to the University of Illinois. I had these really great teachers. I was again exposed primarily to to chemistry at first, but then I got an opportunity to work in a laboratory over summer after my freshman year. In that laboratory, George Ordahl, he was a biochemist, and the work was technically biochemistry, but it was also in bacteria. So we were using bacteria as the tool to deduce biochemical mechanisms that were occurring. And so I learned about microbiology essentially on the side while I was studying primarily biochemistry.
And like usually, like people have a lot of some of the, like when in the career they have somebody who mentors them or like coaches them. Do you have any coaches, mentors, or advisors in your career?
Yeah, yes, many. So it started out that way. George Ordahl became my PhD advisor. His style, his perspective on creating an open atmosphere. All questions were valid questions. Very generous person. He is a very generous person. So he was my first primary mentor on the research side of the coin. My chemistry professor at University of Illinois, Steve Zumdahl, was also a fantastic influence on me. He was real intellect and problem solving and analytical thinking, and I carry those lessons with me. And then as I matured in the program and got to know microbiology faculty, Stan Malloy really sticks out as somebody I'd call him more of an instigator. He's as much an instigator as he is a mentor. He's really prompted me to do things that I at first didn't think I should do. He once told me to, don't play it safe. It's always a good idea to step outside your comfort zone. I sort of took that to maybe to an extreme. I don't know if I've said no to too much recently. So.
Oh, that's good. That's good. Yeah, you had some nice people supporting you and helping you out throughout.
Absolutely, and then that's still the case. So even my current the department chair that hired me here, Mike Apicella, he was a really good influence on me. My new department chair, Pat Schlievert, he's a really great influence. So I talk to these people. I take advantage of everybody that I've worked with. My postdoc advisor, David Zussman, also taught me a lot at the University of California, Berkeley. I learned a lot at Berkeley. I can tell you that much. So, so yeah, you keep you keep in contact with these people that you run into over the years. You build your network. Those people can always tell you, give you good advice, tell you what you should be doing differently. I even have good contacts at NIH and NSF, and consider those people to be in some ways advisory. tell me what's going on
Uh-huh.
at the funding agencies, and so it's a real education to learn from them.
Yeah. And like, have you thought of like any time in your like early on moving to the industry, or just want to stick to the academics? Like, what made you decide to stick to the academics?
I did not really seriously consider industry. I mean, I thought about it,
Uh-huh.
learned about it,
Hmm.
but I didn't consider it it didn't attract me naturally. I didn't want to necessarily go in that direction. I really wanted to be able to explore new things that hadn't been dreamt up. And at the time, I didn't see that industry would necessarily be a vehicle for that. Now, that's not necessarily true. You might there are great research divisions at great companies, but I didn't learn about that until after the fact. So, in some sense, it's just inertia. I went through the system, kept going. You go to universities as a young student. You go through graduate school. You're in a university. You're learning about the university system. It's
Yeah.
very familiar to you, and so that is really why I continued to do it, because it made sense to me, and I liked the concept of being able to develop our own research projects.
Hmm. Yeah, like that's interesting. You never really thought about it, kind of like really kind of interesting in the sense that, yeah, like you are focused on one thing, and over the years you have done a lot of accomplishments for yourself and for others. And like, what really motivates you to do what you are doing every day?
Well, so that and that's what I was going to follow up on it. That's the flip side of that coin is that I've really always just been interested in how things work.
Hmm.
I really like to get to the bottom of what's going on in nature, and
Uh-huh.
I think that's we have to write our grant proposals to to beg for money, and we phrase things that way, but it's actually also really it's really true.
Yeah.
I I love to hike. I love to go out, and I go outside every day,
Uh-huh.
take the dog for a walk, and I'm just constantly making observations, and I just find things to be very curious, and so it's a natural thing for me to want to know how things work. I'm basically interested in the laws of nature, as it were. For that reason, I like physics. I like math. I like astronomy. I like all that stuff, and
Uh-huh.
I think that's what we're trying to do. We are basically interested in getting at the bottom line of how organisms are doing their thing in nature.
Yeah. And what are the challenges? Like, you are into so many things, doing so much of like grant writing, teaching, and mentoring your students and all. Like, what really challenges you face every day?
So all of those things are challenging.
Mhm.
If I only had to do one of those things, I could probably sleep well at night. It's the combination of all of the duties, responsibilities, and the large number of tasks that really makes it very challenging. So managing time, managing people, working with people to get them to do stuff. I can't spend an hour with each person every day,
Yeah.
because that would be 12 to 15 hours right there,
Uh-huh.
and I wouldn't get anything
Yeah.
else done. So I have to I recruit people to the lab who are very independent minded,
Uh-huh.
and we have discussions about what they should be doing, of course, and uh, but we really put people on a path and have them go in the right direction, and
Mhm.
then it's up to them to you know to to blaze their own trail and then come back to me when they get hung up on something.
That
So
is interesting. That is interesting.
Yeah, and that's really how we do it. So as you can imagine, not every project works that well,
Yeah.
but that's also why it's also part of the personality of the laboratory. We have, a great group of individuals, and I can tell you the ones that talk are the ones that do the best, because if something's not working, they talk to everybody else in the lab. They come and talk to me, and we solve a problem, change a project, whatever it might be. People that keep to themselves don't always make it through the hurdles as quickly as they should. So it's a key trait for survival in science, I think, is attempting to be a good communicator, even if you're not necessarily good at it at the beginning. Keep trying, keep doing it. You keep working at communicating with your colleagues. It's a very interactive discipline.
And in addition to what you are talking about, taking dogs to the walk and all, what are the other hobbies? How do you spend your spare time in addition to research, other than research?
Well, if I actually have free time, I like to read, fiction. Basically, I like to get completely off of science. I started out reading technical. I would read a lot of technical books, maybe an economics book or something like that, but it was too scientific. It wasn't terribly relaxing. So I've learned to, you just read literature and just expand my horizons that way. So that's one of my favorite things to do. I enjoy movies, of course. I love art. Try to get to museums, go to Chicago. There's great museums in Chicago.
Okay.
Yeah. But outdoors, it's pretty much the outdoor activities is hiking and biking around town here. We've got a really great trail system. We have a reservoir. The reservoir is frozen right now, but the dog fortunately likes to go for walks on the ice. So we have to go out on the ice and tempt fate there a little bit. So, so we get out quite a bit, 365 days a year.
Wow, that's nice. And tell us about your lab people, like grad student, postdocs, and all. Like,
so so that gets back to that point. When you asked me about the challenges, managing people, managing the projects, managing my time. So I've essentially set up the lab in such a way that we have 3 or 4 projects. Each project has a postdoc. So there's basically 4 postdocs in the lab, 4 graduate students in the lab, and then another fleet of undergraduates. At any given point in time, we have probably 5 to 7 undergraduates in the laboratory. So the postdocs are working with the graduate students are working with the undergraduates, and so there's a cohesive unit on each project. And so that's how it's set up, and it seems to work pretty well. And we have a lot of collaborations, of course, too, because we can't possibly do everything. So we have a lot of collaborations with other faculty in the College of Medicine, in the Department of Microbiology, in the Department of Biochemistry, and Pharmacology. You'll hear about one of those later today when I talk about the Microbiome project.
Okay. And like usually these PhDs, postdoc, they go on for several years. So how do you keep these guys motivated?
They do a pretty good job of keeping themselves motivated. I find that that's a common feature in scientists. They're typically drawn to these, to a PhD program. Usually, you don't need to be motivated externally if you've signed up for a PhD program, and the same is then true for the postdocs. And it's true, though. I mean, projects can get boring or might not be so successful, and that's why we change things up. So it's certainly an adventure. Because we don't know what we're doing, right?
Yeah.
I mean, it's the whole point of research. We just know what we're doing. So it's quite quite a complex puzzle we're trying to solve for everybody's project. And so I think that just inherently keeps people motivated. But we reward people with first of all, they're getting paid fairly well. It's not a bad thing these days to be in science. You can make a living depending on where you live, I suppose.
Yeah.
New York City, you might be just barely scraping by, but here in Iowa City, the salaries are good. People can do pretty well.
Okay.
But I also, like I said, I reward people. They get to go to conferences. The postdocs are going to 2 conferences a year. The graduate students are going to a conference every year.
That's good.
Yeah, sometimes they get to go to more than one because we're local, or some conferences that are closer by that are drivable. So we keep it exciting. Everybody has responsibilities. They have a lot of challenges. I think they're actually just really very busy.
Okay. And coming to the last part, like, what do you like? If somebody wants to join your lab, somebody wants to join as a postdoc or a grad students, like, what is the best way for them to reach you?
So email is definitely the best way.
Okay.
Postdocs, I think every postdoc that has joined my lab, I have met somewhere before. So the best way to get a position in my lab, it's generally something that is organic. We've met at a conference, we've had a discussion. For some, we hit it off. It was an interpersonal relationship. It makes sense that we would try to promote a project together. So that's true for the postdocs. It's similarly true for graduate students. Graduate students come to the university. They go through they do a series of rotations.
Yeah.
So they spend 10 weeks in the lab, and we find out if we get along, see eye to eye on my mentoring style and their needs for advising and their background and their suitability on the project. So it's really a fit thing. It's I don't create a position and then go and then just find somebody to fit that position or to fill that position. It really is a fit.
Okay.
So,
yeah, like if somebody meets in the conference and that's the time to, if at all they are looking for a position and interested in your work,
right,
so they can just talk to you and find out how it goes from there.
That's right. And they can people can contact me, and this has happened too. People have contacted me in advance, meet me at a conference, and then we follow up. So people can find out about what's going on in the lab by looking on the website, getting an idea of what's going on.
Okay, I think we spoke enough about yourself. I think let's talk about your work, and I think you have a pretty good presentation to give right now. I think I'll let you go ahead and start your presentation.
Okay, let's see if we can get this to work here. Okay, so can you see that?
Not yet.
Not yet. Okay. How's that?
Yeah.
Yay. So you can see all the slides on the left?
Yeah.
Okay. All right. Well, we're going to have to do this because of technical issues, but that should work out. So is that pretty good? You're seeing essentially one slide?
Yeah.
Okay. All right. So, all right. So what I'm going to talk about now is this latest project that we have looking at how xenobiotics, or molecules that are otherwise foreign to our cells, disrupt the microbiome and then have deleterious consequences on metabolism. And so I'm going to start out with an intro on the human microbiome, but I want to back up again and say that this is the whole field of the microbiome really took this lesson from microbial ecology, and we've been thinking about what's going on in soil for over 100 years, and with respect to bacteria, and we're really now making similar or great advances using similar kind of thinking for the microbiome, which then is the consortium of bacteria that live in us and on us. So I love these two pictures that I stole from the internet. This picture on the left with a dog giving a baby a big kiss. And it turns out that families that have pets are sharing their microbiomes with these animals, and amazingly, it seems that it's health promoting. And so you might have heard of the hygiene hypothesis, which suggests that kids that grow up in cities that are exposed to a different collection of organisms from the environment aren't necessarily as healthy as those kids that grow up in rural communities. So it's really a good idea. This child on the right is putting things in his mouth and has dirt all over him. And I think the idea is really great that kids like to sample their environment. They put their hands in their mouth. It seems that you're informing your immune system of what's out there, and the more you play in the mud and the dirt, the better off you are. And of course, then we have these amazing numbers that have been determined. There's some debate about that, but on average, a person has about 10 to the 13 cells, and human cells, animal cells that make up our body. But then we have perhaps as many as 10 to the 14 bacteria living in us and on us. That number might be revised downward, but even at the low end, it seems that it's at least 10 to the 13. So we're in a 1-to-1 or 10-to-1 ratio with our microbes. So we're basically a living, breathing, walking, ecosystem. And then to me, and hopefully we'll have time to talk about this, that to me the thing that's even more amazing is that, and what we know very little about, is that there are even more viruses, bacterial viruses known as phage, perhaps as high as another tenfold more with respect to individual phage genomes that exist. And those organisms, the phage, influence the microbial communities, and the microbial communities then influence us. So we really are a complex ecosystem. Okay, so I'm going to give a couple introductory slides on how we determine what a microbiome is, and that's shown here on the second slide. So we're basically doing one of two things. We can sequence everything and determine metagenomes, but the simplest example to give you is the one where the field really broke through, and that's using the bacterial 16S ribosomal gene. And the bottom line is the 16S ribosomal RNA is part of the molecular machinery that translates messenger RNA into proteins. And the 16S gene, in fact, is what was used to create the phylogenetic tree shown at the lower left here, where Carl Woese deduced the existence of a third branch of organisms, the archaea, that are distinct from bacteria and eukaryotes, which include ourselves, multicellular organisms. And so that gene, the 16S gene, it's present everywhere. And so that is depicted schematically up here on the top as this thin black line with these amber boxes. And the bottom line there is that the thin black line is conserved in all organisms, and the amber boxes are variable regions. And so what we can do is simply sequence the variable region of the 16S gene, and that is essentially a molecular fingerprint of the organism that owns that 16S gene. So what we do is isolate chromosomal DNA from an environmental sample, and we can design PCR primers to specifically amplify a specific variable region, such as the variable 4 region shown on this figure 1 here, or on the top of this figure. We PCR amplify that, and then we sequence it, and from that we can create these phylogenetic trees that are shown at the lower right. Can you see my cursor on the screen?
Yeah.
Okay, so these phylogenetic trees then are a representation of the distribution of organisms at a given body site. So we can sample your nose, your mouth. You can get samples, sputum samples, look at samples that come from lungs. You can look at skin all over the body, urogenital tract, intestines, etc. So what we see is that different organisms live in different places. And that's what we're trying to determine across the board for healthy individuals, individuals with diseases, and also looking at what happens over time. And I'm going to show you more of that on this next slide. Okay, so there's too much information essentially if you look at just a single body site. If we PCR amplify material that comes from one of your teeth, you'll have a huge forest of organisms that live there. And so we have to have a good way of representing this data. And so what we do is what the microbiome field has done, Rob Knight in particular, shown on the mentioned down here in the lower right, what he and his team have done is come up with algorithms to compress this information. So you can take a phylogenetic tree and it can be represented essentially as a single dot, which is what you're seeing over here. So now all of these dots are samples from different points in time from a given individual or from different individuals within a population. So we can test everybody that lives in a given area, and we can see how similar or different they are from each other. So it turns out that all these green dots are skin microbiota, and they cluster together because they're more similar than they are to fecal microbiota, which are shown down here toward the bottom. And so now I'm going to try to play this movie. Sorry, that's oral microbiomes. And then there was skin, vaginal, and fecal. And so this is a really great movie that Rob Knight made when he's at Colorado. He's now at UCSD. And now what he's showing is how an infant's fecal microbiome evolves over time. I believe this is about 2.5 years of growth for this infant. And so when an infant's born, their fecal microbiome looks more like the mother's vaginal microbiome. And then over time, it gradually becomes more like an adult fecal microbiome. And that's where you see the yellow dot approaching the adult fecal microbiome. I'll let this movie run a couple times. And then what you noticed there was the child took an antibiotic, and it caused a major shift back towards its starting point, and then it recovered. Can I play that again? I'll just play it again. So we'll run this again so that you get a good idea of what's going on. So different body sites have different microbiomes in the adult, but the infant, when it's born, the fecal samples look more like the adult's mature microbiome at either the skin or the vagina, depending on how the baby is born. That microbiome matures over time. All infants get sick, as you know. They take an antibiotic. Get there in a minute. So this infant takes an antibiotic right about there. Right there. It's a pretty significant shift back towards its baseline, and then there's recovery. But of course, it's also possible that when you take an antibiotic, you may not recover. And that's essentially the hypothesis for our study. We've known from a variety of studies that taking antibiotics can lead to things like obesity. We certainly know that from the farm industry, the agriculture industry. Put antibiotics in farm food and the animals gain weight. Well, it was also noticed that this molecule called risperidone, which is extremely common, highly used, very commonly used antipsychotic in the United States and around the world, causes obesity in children. And our hypothesis is that it's essentially acting like an antibiotic, a xenobiotic, and it's disrupting the microbiome. And this, of course, is based on the work that Jeff Gordon, Ruth Lay, and Peter Turnbaugh have done. And they, first reported in 2008 that you could get, an obese mouse, and you could transfer the intestinal flora, intestinal bacteria from the obese mouse into a lean mouse, and that lean mouse would then subsequently gain weight. So there's something to what's going on with the microbiota, and that's what we were testing here. Now, it's worth pointing out that Johnson & Johnson was sued for $2.2 billion because they knew that patients taking risperidone was during, I think, the phase 2 clinical trials. They knew that there was a side effect of obesity, and they suppressed that information, so they got sued. So this turns out to be something that was probably avoidable, but now the drug is out there. It's being used because it's needed, quite frankly, for psychiatric patients, but yet it has these side effects. And so we are trying to figure out what the molecule is doing, how it's disrupting the microbiome, and then how that leads to weight gain. So what we did was we first presented the molecule to mice. We did a study in humans, but it was a retrospective study. We looked at human samples and saw that there was a shift, but we couldn't determine cause effect because the samples had been collected before we even got involved with the study. So we wanted to do a similar study in mice, and so what we did was we just put risperidone in water, and we feed mice, and we have, of course, control mice in the same room. Each mouse is individually caged. For those who are interested in how we do this, and then we collect the stool, and we sequence the stool, and we also measure the body mass of the mouse. And so what you can see in this graph below is that the mice that were treated with risperidone, those are shown in red here, gain a lot more weight than the growing control mice. And so notice that the y-axis doesn't even go down to 0. So this difference here of 2.8 grams is over 10% of the body mass of the mouse.
That's interesting.
So that's pretty significant. That would be the equivalent of you and I gaining about 30 pounds in 1 year.
Wow.
So this is a huge weight gain for a mouse. Huge weight gain. So it's worth pointing out, though, that just like the infant in the movie from Rob Knight, the infant, when it's born, its microbiome naturally changes over time. The same thing is true for the mouse here, and the mouse is gaining weight. We get the mice from Jackson Labs. They come into the university. They go into the animal facility. They're exposed to the handlers. They're exposed to the researchers in the lab. So they get their own microbiome. It's established when they're born. It then changes when they come here. So we let everything equilibrate. And then the animals gradually gain weight. They mature. They become adult mice. So they gain weight, and when we sequence the stool from these animals, and we create those phylogenetic trees, which again are compressed into these single dots, we can see what happens. So at time 0, at day 0 on this chart, we can sequence the microbiomes, and that's what's shown in these gray and white ovals. So all of the animals in this particular study showed a similar microbiome at time 0. But the control animals, as they gain weight over 2 months, their microbiomes shift, but it's in a particular direction. And these microbiomes are distinct from the starting point, but they're also very distinct from those animals that were fed risperidone. So that you can see that this is essentially an orthogonal shift in a completely different direction to different species, different genera of bacteria.
Mm-hmm.
Okay, so we have a different way of representing that data. Oops. And that's shown here. So as I said, each dot in this display on the left is an individual animal at a particular point in time. Now what we can do, and what we did over here on the right, is collect all of the animals that were fed risperidone and all of the animals that were not fed risperidone at the 2-month time point, and we combined all that data into one diagram. So now you can see the different classes of organisms. Those that are shaded in blue are organisms that are enriched in the controls, and they would represent a healthy microbiome. And those that are enriched in red are the microbiomes that have been disturbed by ingesting the risperidone. Now, we can't say much about any individual, phylum, class, order, family, genus, species. We can't say much about that, but it's the constellation of organisms that we think is contributing to the overall phenotype. So this is just a different way of representing the data that you see on the left. Okay, so then we turned our heads to the question of why. So we can't really say anything about why these bacteria may or may not lead to weight gain. There's definitely a hypothesis out there that comes from Jeff Gordon's work, and the hypothesis is that there is enhanced extraction of caloric potential from food. In other words, you could eat fiber, which we normally think of as passing right through us, but that fiber could perhaps be converted to glucose, and then you could absorb it and you would gain weight from that. So we wanted to test that in this case. I mean, and that hypothesis might be correct in some cases, but in our case, it turned out that we saw that that was not the case. So again, we have the graphical, on the left, we have the graphical representation of the body mass increase for the animals that were fed risperidone. But what we did was we put mice in metabolic cages and we measured the exact number of grams of food that were ingested each and every day. And it turned out that there was no increased intake in the amount of food. We also then did bomb calorimetry on their food and their stool, and that allowed us to calculate digestive efficiency. And you can see there's no change in that. And so basically what that means is the same amount of calories that were extracted from food in the risperidone-fed animals, it was no different from that and the control animals. So the same number of calories are extracted regardless if the animal was on risperidone or not. And so the total amount of energy absorbed is the same. And that means that something else has to have changed. And so we ruled out a bunch of things, and the bottom line is that we concluded that the weight gain must be due to reduced energy expenditure, which means that the animals gaining weight are just not burning calories. Okay. And now that might make sense too. People said, and we thought, well, this could be due to the fact that risperidone is affecting something in the brain, neurotransmitters, the whole brain-gut axis. Maybe there's a whole shift in mitochondrial function, what have you. So we wanted to try to measure this directly. So this is when we started. This is part of the work that we did with Justin Groby and pharmacology. His lab is right below mine. So basically, this is what we did. We used this contraption that Groby built, and it allows us to calculate the enthalpy, the free energy of a mouse. So what we do is we don't just measure oxygen intake and CO2 production, which is respirometry, and that's the usual one of the usual measures to find out if somebody has a metabolic disorder. But we coupled respirometry with the ability to measure the heat production from a mouse. So Justin's lab and Colin Burnett in Justin's lab built this contraption, and I'll show you a picture of it in a minute, where we can put a mouse in a metal box that's thermally regulated. We put a telemeter, surgically implant a telemeter into the mouse. The mouse is happy with that in there. And so we can measure the core body temperature of the mouse, measure the heat that's given off, as well as the oxygen consumption and the CO2 production. And these mice go in the box for a couple of hours. They get bored being in the box. They lay down, go to sleep. So we can actually measure their resting metabolic rate. We take them out of the box, put them back in their cage for several more weeks, a couple of months. They go about their business. They're no worse for the wear. I want to show you the photograph of the apparatus here because it's really fantastic. This is what Colin Burnett built. That metal box is inside the styrofoam container, and so there's a lot of parts here for this. It's there's basically only one entity that is doing this. It turns out they have a similar apparatus on the International Space Station, but if you want to do this kind of work on Earth, you have to do it here with us.
That's interesting.
It is interesting. Hopefully, we'll keep Colin here, keep him busy for a few more years.
Yeah.
Yeah. Okay, so what did we do? So I showed you this graph on the left already. So the mice that were fed risperidone, when we put them in the box and measured their metabolic rate, it turned out that there was no change in aerobic respiration. We were surprised to see that. We were hoping we would see something like this, but it turns out aerobic respiration had nothing to do with it. So it's not your usual, the usual turnover that you think of when you go out and exercise. It had everything to do with non-aerobic respiration. And immediately I started thinking, well, that's bacteria in the intestines. They're not necessarily exposed to, there is some oxygen there, but it's not necessarily a high level. And what you see, though, is a 16% decrease in non-aerobic resting metabolic rate. And that is in perfect alignment with this 2.8 gram weight gain shown. So we can attribute all of the weight gain to a suppression of non-aerobic resting metabolic rate. And that is effectively the calories. Oh my gosh, I should have unplugged my phone. Sorry about that. So that is effectively the calories that you can burn while you sleep, while you're sitting down. It's what you do when you're at rest. Okay, so that was pretty interesting result. Now we went back to the complexity of what's going on with the system here. It's possible that feeding risperidone to the mice is changing something in the mouse, which then subsequently affects the bacteria rather than the other way around. So we wanted to establish Koch's third postulate. Koch's postulate so that if you think you have an entity that causes disease, you have to be able to isolate that organism, give it to an uninfected person or organism, reestablish that disease, and re-isolate the organism. So that's, in effect, what we've done here. Now, this might sound gross, but it turns out that mice like to eat stool. They're coprophagic. It's what they do. And, of course, anybody who has a dog and is taking their dog for a walk knows that they'll eat rabbit pellets and goose poop on the floor. It seems that the animals instinctively do this. Anyway, so we're simply taking advantage of the fact that mice don't mind that. And so we take a control mouse, we isolate their stool, and we give it to a naive mouse. And that's our control. And we do the same thing with the risperidone-fed mouse, and we isolate the stool, and we give it to a naive recipient, and we see if it has any effect. And then we measure the resting metabolic rate, we measure the weight gain, etc. And I want to point out that we, of course, looked in the stool to see if there was any risperidone left, and there was essentially none left. We did a bunch of controls on that. But in the interest of time, I'm just going to go forward on that. And the bottom line is, as you might expect, the transfer led to a suppression of resting metabolic rate in the naive mice that got the stool from the risperidone-fed mice. So we can transfer the phenotype just with the bacteria. So you feed a mouse risperidone, collect its stool, and that stool, when given to a naive mouse, now produces the same phenotype. And it is all due to the non-aerobic fraction. So briefly, this is our conclusion from the study, that the gut microbiota is an ecosystem which positively contributes to metabolic rates. Xenobiotic, in this case, risperidone, disrupts microbial communities in the gut. We know that from the sequencing result, and then since we could transfer it, and it led to the same phenotype. We think that that microbiota can account directly for a portion of the metabolic rate, literally burning calories while you're at rest. Okay, so I have a few more slides that I want to show, and that hopefully help convince you that we've nailed this down. The next part we haven't published yet, but we wondered, can we assay whether or not the bacteria are accounting directly for the change in the metabolic rate? It's still formally possible that the different microbiota is affecting neurotransmitter, hormone signaling in the animal, some other aspect of the immune system, which then affects the metabolic rate. So how do you test it directly? It's not easy. And we've almost got this study ready for submission, but it's not quite there yet. So what do? We took advantage of the fact that mice have this appendix-like entity called the cecum. And the cecum is really just a membrane, an extension of the intestine, a membrane that is like a sausage. It's packed full of bacteria. So the cecum is about 1% of the mass of the mouse, but it's 99% bacteria, and it's just a thin membrane, and there's not much vascularization. And so, like an appendix in a human, you can just cut it out and tie off the stump, and all you've done is remove a little sausage full of bacteria.
Mm-hmm.
So now what we've done is an experiment where we put the mouse in the box, we get the baseline for the resting metabolic rate, we do the surgery. We can do the surgery in about 10 minutes.
Mm-hmm.
Put the mouse back in the box, and we get a new post-surgery, post-removal resting metabolic rate. And the difference between the intact mouse with the cecum and the mouse without the cecum is about 10%. And then we do a sham surgery, noted over here on the right. We do a sham surgery, and it turns out that the before and after are unchanged. So it looks like the metabolic, the 10% decrease, there's 8% in this particular bar graph, this 8% decrease is due to activity of the bacteria in the cecum itself. So we do think that metabolic activity of bacteria is turning over calories, which contributes to our ability to maintain body weight.
Okay.
Okay. Do I have time for two more slides?
Yeah, go ahead.
Okay, so I mentioned this at the very beginning. We're fascinated by the fact that there are lots and lots of viruses, bacterial viruses or phage. There may be 10 times as many phage as there are bacteria in the system. And this nice graph over here on the right from Forrest Rower at San Diego State shows this nice temporal relationship between growth of microorganisms in the ocean, which are then followed subsequently in close temporal proximity with a burst of phage activity. And so essentially what's happening is phage are hanging out in sediment, and then when bacterial blooms come along, the phage have something to infect, and when those phage infect the microbes, you can kill off those microbes, and then when the microbe, the suitable host is no longer there, the phage population also decreases, and so you have this nice cyclical pattern between microorganism and phage, and that's essentially the kill the winner model. So the most successful bacteria then becomes the biggest target for infection by phage. And so phage are helping keep bacterial populations in check in the environment. And of course, we think something similar is happening in the gut. So we did one other experiment, and that is, instead of just transferring the fecal samples to naive mice, we took those fecal samples and we isolated the phage. We separated the phage from the bacteria, and then we transferred the phage into naive mice. And again, we got a similar phenotype. So phage isolated from risperidone-fed mice led to a substantial weight gain and suppression of resting metabolic rate. So that allows us, this last set of experiments, I think really allows us to come to the conclusion that microbiome is accounting directly proportion of the metabolic rate and that phage are affecting the overall balance. So this is we think it's a really important finding, and
Yep.
we're doing a lot of work along these lines to try to find out what else is going on. Okay, so one more slide I'm going to show you, and then I'll probably one more slide and then acknowledgments. We also want to know if we can calculate the delta G of bacteria directly and even for humans. So Colin, who built the big box for mice, has actually built a human-sized box.
Wow.
And it's almost fully functional. And a few of us will be the first volunteers to go into the box. We'll probably have to go there for 3 or 4 hours, fall asleep, and then have our metabolic rates measured. And then we'll either take a supplement that's thought to stimulate metabolism, or something else. We haven't decided what we can do there. We're going to need approval to inject ourselves with anything.
Yeah.
But we've also built microcalorimeter, which is shown down here on the bottom, where we can. These are little copper Peltier setups, and so you can put bacteria. We've done this actually with the cecum. We can take the cecum out of the mouse, put the cecum in here, and measure metabolic rate. But just putting culture of E. coli in there has given us a nice result. So you can see what we have here. When we injected the E. coli, we get a nice raise or a nice increase above the baseline, showing that we can see some metabolic activity. So that's what we're working on. Everything is a prototype in the lab, and we are really trying to go to the next level so that we can measure all these things directly and see how individual molecules may or may not positively or negatively affect metabolism in the host. And so this is my acknowledgement slide. Sarah Barr was the graduate student in the lab that started the microbiome work. I mentioned, Justin is a collaborator, as well as Colin Burnett. I don't know if his name's on here. I have a lot of names on here. But Colin and Justin have done all of the metabolism work downstairs. And we've had just great collaborations, and it's been really terrific. So that's all I'm going to say. Thanks for letting me have a chance to present this. And I'm happy to address any other questions you might have. We'll go back to the conclusion slide.
So if you can, okay.
Should I get out of the?
Yeah, get out of the PowerPoint.
Okay.
Okay, here you go. So
Okay.
I have one question. Like if you take the people or the mouse away from Respirodon for a while, do they lose the weight, or it still remains? Is it a transitionary or it is permanent?
It is. So the correct answer is we haven't done that experiment fully to establish that. We've done some of the preliminary work, which is take the Respirodon away, and we see some recovery. But what we haven't done is the prolonged time course yet with this sequencing to show that the microbiome returns absolutely to baseline, and it might not.
Yeah.
Yeah, it might not, because these mice are, we expose them to. Sorry about that. We expose these mice to. We expose the mice to the molecule for 2 months, and they're continuing to gain weight, and so we haven't. It will probably take many more months to do that experiment properly. We are doing a series of experiments similar to that, which we think will help us determine that. So, but the question is a good one. Can we displace the unhealthy microbiome with a healthy microbiome? Can we? And that's really where the whole field wants to go here. Can you develop a probiotic that would then help generate health in people? So we want to do that. We just haven't done it. It's probably going to take us a couple of years to do that right.
And is there any species different mouse, different rat do behave differently because of their gut microbe maybe slightly different?
Can you ask that again?
Like, is there any species difference, like different mouse,
Oh,
differently bred, different varieties?
yeah. Okay, that's a great question. Yes, absolutely. So we're using the C57 black 6 mice, and it turns out they happen to be great for these studies for weight gain.
Uh-huh.
They're very different. The different strains, the different lines of mice can behave very differently. They'll respond differently to the same antibiotic. And we haven't done that assessment. We haven't ordered 10 different strains of mice to test them all, just because it's very expensive.
Yeah. So why I'm asking is basically people eat differently from different parts of the world, have different kind of microbiome.
Yes.
So is it true for everybody? It is kind of a general phenomenon or not? Kind of.
No, I think that's right. I'm pretty sure that's going to turn out to be right. Yeah, I mean, there's a nice study that came out of Israel showing that multiple diets all lead to a healthy gut microbiome, but they're different from each other.
Yeah.
So that's a really good point, and allows me to bring up one of our future goals here is to see if we can map out that sequence space, if you will. What is healthy and what is not healthy. If you remember that movie from the beginning, there was a lot of there's a lot of distribution, even though things are clustered based on body site. There's still a lot of distribution, and that's certainly the case. We share a large amount of our microbiomes with the people we live with our family members. They're not the same. The host is also selecting for which organisms live there. And if you have eczema or psoriasis, you have different microbiomes, in different parts of your skin. So the host's genetic determinants also affect that. So we will. No doubt, we're all going to have a huge heterogeneity there, but nevertheless, we think we can probably identify a core microbiome that might be pro health.
Yeah. Okay. I think we are almost ran out of time, and thank you very much for taking your precious time, talk to me, and being here. And we'll see you, if you don't mind, in few months when you have more results. I'm really interested in that human experiment.
So am I, because I'm probably going to spend a weekend in
yeah. So, let me know how it goes. It may be something really interesting result. So certainly we would like to learn more about it. It's really looking like what we want to learn, what we want to see, kind of neat experiments you are doing.
Yeah, and it's fun. So that's what's keeping me motivated right now. Fun sure. So thanks a lot for having me. It's definitely fun for me to talk about it.
Yeah, thank you again. So we'll keep in touch, and we'll see you again next time.
Okay, thank you.